Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is for the discussion aspect of Lab 15 and 16, the Sensory System Lab. So, quick review of the experiment video. We have many parts within this experiment. For the test, for the exam, the quiz, I do expect you to know what's going on in each one of these parts. I want you to be able to give a general description of what's happening in each one of these things and the science behind what's happening in each one of these experiments. So we have mapping of skin receptors. This is where we drew 16 dots on our forearms and we touched each dot with a room temperature probe and then a hot probe and a cold probe, 2-point threshold, which is where we started with the anesthesiometer, where we had two prongs that were right next to each other, and we poked our fingertip. We spread it 1 millimeter apart, poked our fingertips, spread it 2 millimeters apart, poked our fingertips, and we saw at what point did we feel 2 dots, 2 points. Receptor adaptation, this is the one where we put one hand in warm water and one hand in cold water, and then we put them both into a bath of room temperature water, and we saw how it felt. Referred pain was tapping on our funny bone or cubital tunnel. Acuity, we did 2 different things for acuity. I do want you to know both things. For acuity, Snell and I chart and 2-line discrimination test. Then we did the astigmatism and accommodation labs, or parts, yeah. So, results. The results for cutaneous sensation are up here. Generally, every student will feel all 16 for touch, or pretty close to it. There might be 1 or 2 that some students miss, but usually pretty much all of them students feel. With cold, there's usually some students that feel all of the cold probes, and I think that's mainly because of how hard they're pushing down on their skin with the probe. But most students, they're missing about half. They're missing about half of the dots where they don't feel cold at all. And with warm, it's usually about half to about 3/4 of the dots the students feel the warm sensation on. What does this all mean? Well, the receptors that detect touch are different than the receptors that detect cold, and the receptors that detect cold are different than the receptors that detect warm. Different receptors for different sensations. Different receptors for different sensations. There's not just one temperature sensor. There's cold sensors and there's warm sensors. These receptors are physical things. They are physical things. You can look at them with a microscope. They're physical things. And where they are located, there are some spaces where they're not located. I mean, think about everything there is. There's skin cells, there's other receptors. Every single part of your forearm does not have cold receptors on it because it's got to make room for warm receptors. It's got to make room for touch receptors. It's a physical thing that is not ubiquitous across your forearm. The density and location varies. Density and location varies. Touch tends to have the highest density, which is why students usually feel all 16. Cold and warm usually have a lower density for those receptors. For cutaneous sensation, so for this one, you can see the results right here. This experiment, it's easy to do at home with a paper clip. You can unfold a paper clip, get the 2 pointy ends, and start with them kind of right next to each other and slowly spread them apart. And you'll notice when the 2 points are right next to each other, even on your fingertip, you can't tell that it's 2 different points. But as you spread it apart to 1 millimeter, 2 millimeter, 3 millimeter, you'll start to feel that they're 2. And you'll notice that this number, how far apart they need to be, is different for different body parts. It depends on receptor density, size, and shape. So I've got a question for you. Receptor density, how many receptors there are per square centimeter? The higher that number, the more receptors there are, the finer your ability to feel things are. Is it important to have a really fine sense on your fingertips? Absolutely, because we're humans, we make tools, we move things with our hands. Is it important to have really acute, really accurate sensation on the back of your neck? No. I mean, it's like, oh, I feel a spider falling on my neck. I need to get it. It's not like you need to feel exactly what's going on there. You're not moving things around with the back of your neck. So different parts of our body have different densities of receptors. Fingertip has the highest density. Palm of the hand, also important for making tools and being a human. 6 millimeters is the average 2-point discrimination. Back of hand, 9 millimeters. Back of neck, 19. If you go to the next one, look at this, calf. That's like 5 centimeters, 6 centimeters. That's a really long distance. All right. Perfect. Cutaneous sensation. So adaptation of temperature receptors. Adaptation of temperature receptors. So who has ever been in a hot tub and then got brave and ran and jumped into the pool? So I've done that. I think a lot of people probably have. When you're in the hot tub, your temperature receptors adapt to that warm temperature. Your temperature receptors adapt to that warm temperature. You then get out of the water and jump in the pool. And the pool is usually like normal pool temperature. Like it's a fine temperature. But because your temperature receptors are used to that warmth, the normal temperature pool feels really, really, really cold. That's what we're doing in this lab. We start off with one hand in warm water, one hand in cold water, and we let our receptors adapt to those two different temperatures. We then place these hands into a room temperature water bath at the same time. The hand that was in the warm water adapted to the warm temperature. So when we put it in room temperature water, it feels cold. The hand that was in cold water, we put in room temperature water, it feels warm. It feels warm because it got adapted to the cold water. Here's another thing about temperature receptors. Temperature receptors don't detect absolute temperature. They detect relative temperature. So what do I mean by that? What temperature is it exactly where you are right now? Oh, here in San Francisco, it's exactly 62.354 degrees Fahrenheit. I can tell that because that's what I feel with my temperature receptors. No, that's not how it works. Absolute temperature is like putting a number to it. It's like, I feel that it's 64 degrees right now. I feel that it's 72.43 degrees Fahrenheit right now. No, you don't feel that. You feel hot or cold. You feel comfortable or hot or warm or cold, chilly. You feel relative temperature. So your receptors adapt to the warm water with your hand in warm water. You put it in room temperature water, and your receptors just know it's colder than the water it was just in, the water that it just adapted to. Your hands are in cold water. You move it into the room temperature water. It doesn't know that it's room temperature water. It just knows it's colder than what it's used to. It knows that it's relatively colder. Different receptors have different speeds of adaptation and ability to adaptation. Touch and pressure receptors tend to adapt quickly. Now, you're probably going to do this when I say it, but before me saying this, do you feel the clothes on your body? Do you feel your socks on? Do you feel your shoes on? You have to think about it. Your receptors get used to wearing shoes, It's not like shoes are rubbing against your feet the whole day. It's because those touch and pressure receptors get used to it. They adapt. If something new brushes against your arm, though, if you're on a hike and a branch brushes your arm, you feel it. It's a new stimulus. Your arm hasn't had time to adapt. You know, are you feeling the pressure from the chair you're sitting in? You know, maybe if you think about it or right when you sit down, but not after your receptors adapt. Receptors that are a little bit slower, so temperature receptors are a little bit slower. In this experiment, we kept our hands in the water for a minute to allow them time to adapt. Proprioceptors. What are proprioceptors? Proprioceptors are receptors that tell you where you are in space. So, where is your hand? Where is your finger? Where is your elbow? Proprioceptors help you know where your body is. You know, like if there was a line drawn on the floor and you closed your eyes, could you walk along that line? You know, it tells you, like, if your eyes are closed, you're not looking at your body, where your body is. They tell you, yeah, where your body is, where your shoulders are, how your posture is, where your arms are. And those are a little bit slower to adapt. Never adapting, pain receptors. So pain receptors, for better or worse, don't usually adapt. Pain receptors or nociceptors is another term for pain receptors. Nociceptors are pain receptors. So why would it be a good thing that pain receptors don't adapt? So, you know, if something's hurting you, you know you need to do something about it. You don't forget that you need to do something about this thing that's hurting you. Why is it bad? Well, because you're going to have patients where they're in pain all the time. Like, they're in pain in perpetuity. You know, you ask them when their pain started and they'll say, oh, well, back in 1992, this happened, and they've been in pain ever since. That's not a good thing. And yeah, you're going to have patients that have that issue. So, you know, it's good in some ways, it's not so good in other ways. Cutaneous senses, referred pain. So referred pain is very, 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 very important for healthcare. Why do people go to the doctor? Why do a lot of people in this class want to be nurses? Why do people go and seek care? A lot of times it's because they feel pain and they don't like feeling pain. Referred pain is pain that is felt that's not arising from its origin. Pain felt not arising from its origin. So pain that is felt somewhere that is not where the tissue insult is, or the damaged tissue. So common ones that people know about would be if you hit your funny bone. So what we did in this lab, we tapped on our funny bones. Most people have bumped their funny bones before, which is their ulnar nerve. And they feel pain shooting down their arms into their pinky finger. Well, that's referred pain. You hit your elbow, but you feel it in your little finger. You feel it along the medial aspect of your forearm. Referred pain. It's not just when you damage nerves, though. Heart attacks or angina. It's kind of just common in our society, I think that most people have heard that when you have a heart attack, you kind of feel pain in your chest and pressure in your chest, and then you can have pain going down your arms. Well, the problem is not in your arms. The problem is your heart. You know, some people feel pain moving into their jaw or arm or back. Again, the problem is not in your jaw, arm, or back. It's you're having a heart attack. Pain not felt where the origin is. Brain freeze. You get your mouth cold and you get a headache. Referred pain. Referred pain happens all the time. It's super, super, super common. And you will have patients that have referred pain. In class, I guess we can do this here online. Most students have really tight trapeziuses, levator scaps. Find a tight spot in your levator scap or your trapezius and push down. I know when I do it, I kind of feel pain like coming up the back of my neck and like up to like the top of my ear. You can cause referred pain. Ooh, yeah, I feel it right there. I feel pain where I'm pushing, and it's kind of moving up behind my ear. So pain is not always felt where the tissue damage is. Really common. So right here, pain is a conscious response to nociception. Nociception, so nociceptors sending signals. Can consciously interpret pain when not there. So ultimately, pain is in people's mind. Now, don't go telling all your patients, oh, the pain's just in your mind, suck it up. Pain's a very real thing that people experience. Ultimately, though, it's the interpretation of it in their minds. And that mental influence can enhance or dull. Some people who have chronic pain, their mind becomes used to that pain. And so they just continue to have it, and they continue to feel it even after the issue is gone. Phantom limb. So this person right here is lost part of his arm. Phantom limb syndrome. People can feel sometimes lost limbs. They can feel their arm or pain in their arm when their arm is literally not there. Another example of pain as an interpretation, when my first girlfriend dumped me, I felt pain, like it physically hurt. Now, did she beat me up? No, she dumped me, but like I felt physical pain, mental influence. Pain is something that you experience. It's something that ultimately it's an interpretation in your mind. Pain's a very, very complicated thing. Pain science and the study of pain, there's a lot of arguments in it. There's a lot of things that we still don't know. All right, congenital analgesia, people who just can't feel pain. People who literally just can't feel pain. All right, acuity. So acuity is your ability to resolve objects, your ability to have a clear picture of an object. So Snellen eye chart, you should be familiar with this terminology and a brief description of how it works. A little bit about interpretation of the Snellen eye chart. So I expect you to be able to do this for the quiz and for the checkup. 20/20 vision is something that most of us have probably heard of before. What 20/20 vision means is at 20 feet, you see what the average person sees at 20 feet. So how far away do we stand for the Snellen eye chart? We stand 20 feet away. That's the standard. The average person doing the Snellen eye chart can read line 8. The average person can read line 8 on the Snellen eye chart from 20 feet away. So that's 20/20 vision. If you can only read line 7 from 20 feet away, that's what the average person can read at 25 feet away. So that's 20/25 vision. Now it always starts at 20. So 20 something. At 20 feet, if only you can only read line 2, at 20 feet, you can read what the normal person, the average person reads at 100 feet. At line 3, at 20 feet, you can read what the average person can read at 70 feet away, and so on. So DMV blind is 20/40, blind, legally blind is 20/200. 2-point discrimination test. So this is the one where we have two lines a millimeter apart. We cover one of our eyes, we stand next to that, and we walk backwards until those two lines become one line. Until when we cannot discriminate, we cannot make out both lines. You can do some math that's pretty interesting with the 2-line test. So the distance 1 is the length of the average eyeball, 25 millimeters. Distance 2 is the unknown width of 2 cones in the back of the retina, so X for D2. Distance to the wall is how far away you measure it, how far away you can go without that point where the lines become blurry. D4, distance 4, is the 1 millimeter distance between the lines. If you punch in these numbers, and we do in lab, in class, we come up with our own numbers, this unknown distance with the two cones, we actually calculate pretty close to the real value for most students in class. It's pretty fascinating. Astigmatism, so an imperfection in the field of view which prevents light from passing equally through all parts of the eye. So for some reason, eye does not travel equally through all parts of your eye. You look at this wheel right here, and you'll see differences amongst the length, the width, the darkness of some of the lines around it. Light has to pass through your cornea, the aqueous humor, the lens, the vitreous humor. There's a number of different structures it has to pass through, so it's not necessarily a diagnosis. It just kind of tells you that something's not perfect with how the light passes through. Astigmatism. For accommodation, accommodation is focusing on things from different distances. So like your camera, when your camera focuses on things that are further away or closer, it changes the shape of your lens. Your eye has a lens also, and it has to change the shape of the lens on your eye in order to focus at things at different distances. Your ciliary muscles pull on your lens and change the shape of your lens. As you age, your lens becomes less pliable. As you age, your lens becomes less pliable. You can't change its shape as easily. So your ability to focus on things close to your eyes diminishes. You know how, like my dad, when we go to like a restaurant and it's got tiny letters, like he has to hold the paper, he has to hold the menu, like 2 feet away from his head or 3 feet away from his head if he doesn't have his glasses. It's because his lens is not pliable enough to focus on things close to his eyes. So what we do in class is we find out our near point. The near point is how close you can focus on something. How close can you get an object with it not becoming blurry? That distance in centimeters is your near point. Typically 10-year-olds, their near point is 9 centimeters. 20-year-olds, their near point is 10 centimeters. So let's take Joe, for instance. Joe is 53 years old. Where do you expect his near point to be? So Joe's 50 years old. Where do you expect his near point to be? About 53 centimeters. Let's say Cindy Lou is 30 years old. Where do you expect her near point to be? 30 years old, 13 centimeters. Let's say Marco is 55 years old. Well, where do you expect Marco's near point to be? Well, 55, we don't have an exact number for 55, so it's got to be somewhere between these two numbers, somewhere between 53 and 83. Let's say Julie is 20 years old, but her near point is 18 centimeters. Her visual age is 40, because 18 centimeters correlates with an average age of 40, even though her chronologic age is 20. Julie's 20 years old chronologically, but her near point's 18. Her eyes are as pliable as a 40-year-old's. All right. If you have any questions, please email me, and I'll see you in the next video.